Chapter 3, Cell Structure and Function. We're going to start off this chapter with a brief overview of some of the properties of living systems and living cells and talk a little bit about the cell theory of life. In the second part, we'll talk about the typical parts that a prokaryotic cell might have, and this can be used to help us to identify different kinds of microorganisms. And finally, in part 3, we'll talk about the different parts that a typical eukaryotic cell will have. As always, I like to start off each chapter with a explicit set of learning goals that the typical student should be able to master as they go through and read and understand Chapter 3. By being able to cover these learning goals, you should be able to, at the end of this, answer the vast majority of the questions at the end of Chapter 3. Some properties of cells. The idea of the cell theory of life was first originated by Robert Hooke. You can see here in this picture. He was an English scientist from the 17th century who proposed the idea that all living cells originate from other living cells. What we now know as the cell theory of life. Since then, we've found that all living systems, all cellular life, shares some basic properties in common with one another, regardless of the type of cells that they are. All living cells will increase in size as they grow. Living cells and organisms, if they're multicellular, have the ability to reproduce and make copies of themselves. This reproduction can happen by sexual means, can happen by asexual means, and in asexual reproduction, an exact copy of the cell is produced in that process. Living cells and organisms will respond to changes in the environment. And then finally, all living systems, all living cells have a basic property of metabolism. They take up nutrients from the environment around them and use those nutrients in controlled chemical reactions to accomplish cellular work. Prokaryotic cells are typically going to be rather small, about 1 micrometer to perhaps 5 to 10 micrometers in diameter, with a relatively simple internal structure. Even though they're rather simple inside and lack internal things bound by membranes, we'll find is that actually there's a lot of stuff going on inside them, structures that we can identify using the microscope that will facilitate their identification. Prokaryotic cells are broadly composed of 2 classes of organisms, the bacteria, the archaea, which superficially look very, very similar to one another if we were to happen to see them under the microscope. However, genetically, they're incredibly distinct from one another. Eukaryotic cells, by contrast, are much more busy inside, with a lot of internal membrane-bound organelles. Their nucleus is surrounded by a membrane, typically much larger, anywhere from 10 to 100 micrometers in diameter, with this very, very complex and rich structure inside. Even though these cells are quite complicated inside, we find that all eukaryotic cells, regardless of the type of organism that they come from, are actually extremely similar to one another. And even though algae, protozoa, fungi, animals, and plants look incredibly distinct from one another, at a cellular level, they're very similar to one another. Here we see a couple of examples of different kinds of cells, and our first clue as to what each cell might be is actually given to us by the size of the organism. Look, for example, at these orange cells up here in the upper left. We can see the length of these cells is probably approximately 2 micrometers in diameter. That's a good indication that the organism up here are prokaryotic cells. Over here in B, we see actually 2 kinds of cells, these and this much larger one. We can see that the larger one in diameter is probably going to be somewhere on the order of around 15 to 20 micrometers in diameter. This is a neutrophil. It's a human white blood cell. This red blood cell is also a human cell, but you'll note that it doesn't have a nucleus. And in fact, human red blood cells are the only cells in the human body that lack a nucleus. Over here in C, we see a very long cell. If we were to gauge its length, we'd probably guess that it was probably on the order of around 200 micrometers in diameter. This is almost certainly a eukaryotic cell. And then finally in D, a humongous organism. Looking at the diameter of this, we would guess that this cell right here is probably on the order of around 10 millimeters in diameter, which would certainly be easy enough to see by the naked eye. prokaryotic cells, as we said, are typically rather small, and eukaryotic cells can typically be much larger than prokaryotic cells. The very largest of the eukaryotic cells are oocytes, and we can see that this ostrich egg is around 13 centimeters in diameter. In contrast, a bacterium that we're going to see a lot of in lab, Staphylococcus aureus, is only about 1 micrometer in diameter. The reason that eukaryotic cells can get much larger than prokaryotic cells is because they have these internal membranes, these organelles, these structures inside them that in essence increase the amount of surface area that they're able to utilize. How's that work? Let's look at this cartoon. A table here looks at a couple of hypothetical cells, and here I've drawn 2 hypothetical cells. We can see right here a rather small round cell and a larger round cell. And if you consider that each of these cells has to take up nutrients, think where the nutrients have to come from. They have to come across this surface right here. And the larger a cell gets, the surface area doesn't increase at the same rate. So nutrients entering without internal membranes, what that means is that the larger cell down here is not going to be able to grow as quickly. Now consider what happens when we take that larger cell and give it some internal membranes, some organelles inside. As you can see here, the internal membranes right here have increased the surface area. The surface goes up here like this, goes in and back and around and then back in over here. And by increasing the surface area, even though the diameter of this cell is exactly the same as it is over here, what this means is that nutrients can get into the cell, the big one over here on the right, more easily because of the internal membranes. What that means is that the bigger cell will then be able to grow more quickly. Okay, now here's an overview, a listing of the types of prokaryotic cell structures that we're going to go through and cover. We're going to, as we do this, work from the outside of the prokaryotic cell in and talk about each of them in turn and what they do for the various types of prokaryotic cells that may have them. Here we see a cartoon which sort of illustrates the internal structure of a prokaryotic cell. You can see that the largest portion of the cell is actually made up by this kind of amorphous section here known as the nucleoid. This is actually the genome of that prokaryotic cell, the DNA of it. It serves much the same function that the nucleus does that you've probably heard of in eukaryotic cells. However, there's no membrane surrounding it. Other structures in here, the inclusion bodies, the glycocalyx, the ribosomes, we'll talk about in turn as we go through this. The first structure that we're going to talk about that's found on some but not all prokaryotic cells is the glycocalyx. The glycocalyx, or sugar coat, actually comes in 2 different forms depending on how tightly associated the glycocalyx is to the surface of the microorganism. If the glycocalyx is rather tightly associated and firm up against the surface, we refer to that structure as a capsule. If it is looser and less tightly associated to the surface, we refer to it as a slime layer. Irregardless whether it's a capsule or a slime layer, they are really trying to do the same 2 things for the microorganism that's able to possess them. First, it helps to protect that microorganism, and second, it helps it to attach surfaces. When we talk about protection, we usually talk about protection of that microorganism from things that might try to destroy it, namely things like white blood cells through the process of phagocytosis. A microorganism that has a capsule or a slime layer is able to get away from these things that might try to destroy it. The second structure we're going to look at is an optional structure found on some prokaryotes, but not all, and it's called the flagella. The flagella is actually a rather complicated structure that attaches to the surface of the cell via a basal body that is found right down and crosses across the membrane. The part that we actually want to focus on is the part that's outside of the cell, and you can see that it kind of truncates right here, but it's actually very long and will extend away from the body of the cell. And it's mainly composed of a protein called flagellin. Flagellin polymerizes and forms the large flagellar structure. The other protein that's very, very important to the function of a flagella is found actually down in the basal part of it, and it's an enzyme called ATPase. And as you might surmise, what ATPase does is it carries out this chemical reaction that we see right here, where ATP is hydrolyzed to form ADP plus free phosphate. And of course, during this chemical reaction, energy is also released. The chemical energy of ATP is released by the action of the enzyme, and that chemical energy is converted into the energy of movement. And what ends up happening is that this flagella begins to spin right around the basal body in a motion, a circular motion, a rotational motion that allows the flagella to move. And then what happens with the microorganism is the flagella actually acts as a little tiny electric motor. Here we can see 3 different kinds of bacteria that possess flagella. Some microorganisms have a number of flagella all around the surface of the organism, around the surface of the cell, an arrangement known as peritraceous. Some microorganisms will only have a single polar flagella, whereas some other organisms will have a number of flagella, but they all come to a single point. The number and arrangement of flagella is unique to each species of microorganism, and the arrangement of flagella can help us to identify what that organism is. Certain spiral bacteria called spirochetes have a single flagella, but the flagella is not immediately obvious when we look at the microorganism under the microscope. The flagella is actually very tightly wrapped around the surface of this microorganism. It's wrapped and becomes actually part of the surface of this microorganism, but it does the same thing. ATPase causes this structure to spin, and it spins along the surface of the microorganism, and this results in a corkscrewing motion as this microorganism begins to move through its environment. The movement of a microorganism through its environment can be in response to a number of different chemical cues, this idea of response to things out in the environment that all cellular life is able to exhibit. If we talk about an organism moving towards or away from a chemical, we refer to this process as chemotaxis. Organisms may be attracted to certain chemicals that perhaps the organism might want to obtain more of. We refer to this process as chemoattraction. Likewise, some microorganisms may be repelled away from certain chemicals and move away from them. We refer to this process as chemorepellent. Organisms can be responsive to things not chemicals. They may be responsive to things like light. We refer to this process as phototaxis, and certain organisms may move towards or away from a light source depending on whether or not they might use it. The movement of microorganisms is actually rather random. One process that goes on is this process of tumbling that we can see over here on the left, in which case the flagella on the surface of the microorganism spin in essentially random directions from one another. This has a result that the microorganisms begins to point randomly at different directions in the environment. This tumble movement is alternated with running movement, where all the flagella spin in unison, propelling the organism into a single direction. As the organism moves towards a chemoattractant, the number of runs becomes more frequent than the number of tumbles, resulting in the movement of the microorganism gradually towards the attractive chemical. Here we see a new optional structure. This microorganism here has flagella, these very long structures that help it to move, but there are these other much more shorter structures called fimbriae that superficially resemble flagella. However, fimbriae are not important for the movement of this organism through the environment. Organisms that possess fimbriae are actually going to be able to attach to surfaces together. So we actually now have 2 different types of structures found on some microorganisms, the fimbriae and the Glycocalyx, both of which enable this microorganism to be able to attach to a solid surface better. An organism that has both fimbriae and a Glycocalyx, either a capsule or a slime layer, is going to be able to attach better than an organism that has neither one of them. Here we see a third structure that can extend from the surface of some microorganisms, the pilus. The pilus doesn't help the organism to move and doesn't help it to attach, except in some sense it helps this microorganism to attach to this one. What the pilus enables a microorganism to do is to be able to exchange genetic information. Let's say that this bacterium right here has a trait that might be useful for it. For instance, perhaps it's resistant to a given antibiotic. That antibiotic resistance ability, that phenotype, is going to be due to a certain segment of DNA that's found in the genome of this bacterium. That genetic ability, that genetic determinant, can be transferred through the pilus to the recipient bacterium. This bacterium would then be able to acquire that ability to resist the antibiotic, and that by this mechanism, antibiotic resistance can spread through a bacterial population. You can imagine then that a bacterium that cannot form a pilus is going to be unable to transfer genetic information. It's going to be unable to transfer DNA, and consequently, an ability such as antibiotic resistance might not spread through that population as rapidly if the ability and the function of the pilus is blocked. One last topic to cover before we start talking about some structures inside the cell is the ability of a microorganism to form a biofilm. We've seen two structures that enable microorganisms to attach to surfaces, and this is a key first step in the process of forming biofilms. Microorganisms that are unable to attach to a surface will be free-flowing in a liquid environment. We refer to this as planktonic growth. But if the organism is able to attach to a surface, perhaps by having the structure such as a Glycocalyx or fimbriae, attachment does occur. The microorganisms adhere to a solid surface, and what they find then is that then some of the properties of the organism become very, very different. Certain genes are turned on, other genes are turned off. Consider, for instance, a microorganism that's attached to the surface is not going to need to move at all. The biofilm structure, as these organisms begin to grow and multiply, gets larger and larger and larger and larger until we have finally a mature biofilm, a true community of microorganisms. What are some of the benefits of being able to form a biofilm? Well, if you consider a microorganism that is able to attach to a very advantageous location, perhaps one that has many nutrients, it's going to offer the organisms within the biofilm a very ready, a very available source of that nutrition. An excellent example to think about this is in the human mouth, where microorganisms attach to the surface of the teeth and the tongue and the gums. Humans, as they typically do, will put food in their mouths on a pretty frequent basis. So a microorganism that is attached to the surface of the tooth is going to have a pretty convenient source of food as they remain in that environment. Organisms that are associated in biofilms are able to cooperate with one another and remain in favorable environments. One other very interesting thing that we see about microorganisms that are able to form biofilms is that they typically become much more resistant to antimicrobials, antibiotics, and various chemicals that we might use in order to try to keep them under control. Microorganisms in biofilms over here will be much more resistant to these types of chemical warfares that we use against them than the same species of bacteria growing in planktonic growth.